presentation. So the first presentation this afternoon is titled Biosecurity, Helping Your Plants Thrive. And this will be delivered by Dave Van Wellehem. Dave graduated with a Bachelor of Science in Agriculture from the University of Manitoba and continues to enhance his industry knowledge through ongoing education. He has spent his career working in various production animals units in a variety of roles, from hands-on to managing units to service representative to veterinarian assistant. Dave is currently the National Biosecurity Specialist for Vitoquinol Canada, where his education, practical knowledge, and hands-on approach allow him to bring a unique perspective to biosecurity that is easy to understand. So I'd like to welcome Dave, and I'll hand over the controls to you, Dave.
Thanks, Kara. I got the presentation right up here on the screen, and we're going to get right into it because we got a limited amount of time. So when we talk biosecurity for plants and all that, people always think about biosecurity as more as an animal thing. But if you look at the definition of biosecurity, biosecurity is animal and plant health sectors to help protect them from biological resources, from unintended exposures of pathogens. So we're going to talk about that a little bit today. And first thing I want to show you is that biosecurity is a huge field, and I'm only going to tackle a little tiny part of it. I'm going to actually tackle the cleaning and disinfecting part of it. But you can see there's tons of different areas right now that are also included in the biosecurity. And this is a living science. So it's always adapting and changing as we learn more and more about different diseases and set up new type of programs and so on and so forth. So first of all, I want to point out that under the cleaning and disinfecting, Vitoquinol actually has a biosecurity website, as you can see on the screen, the URL. Right here, lots of stuff we're going to be talking about will be on this website, so you can actually go to it and see them after you're done the presentation today if you're curious. So let's start right in. So on the actual website, this protocol is there, and these are basically steps we're going to talk about: dry cleaning, then cleaning, removal of equipment, detergent application, whether it be spraying or foaming. Follow that by rinsing or washing with high pressure, disinfection, that's a liquid spray-on disinfection, and then finally following up with aerial or fogging disinfection. So those are the basic steps that we're going to go through, and those are the basic steps of any kind of a washing procedure. First of all, removal of organic matter, that's really essential. High levels of soils and leftover matter and stuff like that are great environments for any kind of pathogen to survive and multiply. So we want to try and get as much of that away as possible. And we know that any kind of detergent or even disinfectant cannot penetrate a huge amount of organic matter. So we move as much as we can. If we look at it, it looks actually fairly clean. We're actually quite surprised once we take most of the litter away, it looks. But if we really look closely, we can see there's a lot of these little films that are left on, or inside pipelines or stuff like that that we couldn't actually physically clean. And these are what we call biofilms. And inside the biofilm, there's a lot of pathogens that hide. So I want to talk a little bit about biofilms first. So what a biofilm is that the pathogen, whether it be bacteria, fungi, or whatever it happens to be, will fall to a surface, whether it be inside a water line or on a surface or whatever it happens to be. It'll secrete a material that's a really sticky matrix, and underneath the matrix, it'll start growing because it's very protected, because nothing can penetrate that matrix that's up there. Once it keeps growing further and further and further, it gets so big that it actually breaks apart and starts in several new locations. So you can see that it can be a real hazard as it grows further and further, and it's very quick reacting and building. And this cannot be penetrated by any kind of disinfection, and actually physical manual labor or manual washing doesn't actually take off the biofilm. So in order to take off the biofilm properly, we have to use a detergent, which we're going to talk about in the next little bit. So when you talk about this kind of a film where you see the organics, slime and all that, this is something that we're normally challenged with the most. And in order to get a properly organic deposit, you need to use what's called a degreaser or a high pH detergent. Our example from our company is called Biosol Plus. But every once in a while, this is what we're going to use most of the time, but every once in a while, a rotation is important because when we use any kind of a water against wash something, a little bit of a mineralizing is left. And as you can see on a lot of these pictures here, you can see the rusting look or the dulling of metals and stuff like that. So every once in a while, every fourth or fifth wash, we need to change the type of soap to descaler or a low pH. Our example is Biosol AFC. But let's start before we start washing and cleaning the surface is actually to look at the water systems. And the reason why I'm talking about water systems first is there you'll see in a few minutes. You can see there's a lot of different microbes and pathogens that are possible in staying in water and growing in water, especially inside those biofilms. So it's really important to get a hold of them first, so that because that's one of the main things we feed our plants is the extra water. So the best way to look and see what you're dealing with is actually to split the lines and look and see inside there. If you see a slimy little part of things like this is what we call more of organic. If you see a lot of hard, gritty type of things, that's more of a mineral. So that's how you can tell which way you go. Now you can go a lot of different ways. You can take water samples and swab samples and actually have chemicals that you can test on site. But usually splitting and looking and feeling is actually the more efficient way to go about looking at it and checking what you have. So in order to properly clean a water system, it's really easily to prepare a stock solution to ensure that the water gets into all the pipes and all the delivery systems. And you can go to our Biosecurity website and actually can help you figure out exactly how much product you need. So ensure the product's everywhere by checking the end of water lines and so on and so forth. And then again, Biosol Plus if you got the organics, and Biosol AFC if you got the mineral. Let that stand for a good amount of time so that you can so it can soak. And that's why I like to talk about that first, because it can soak for hours and a matter of fact to days while you wash these surfaces. Once you're done, you can rinse the line and you can see the minerals coming up in here and you can see the organics on the bottom. Following that, you can disinfect the lines again with a proper disinfectant. And these don't have to sit in for days. They can just sit in for hours before you flush them again, before you start your new crop of plants. Now, if we look at the surfaces, it's the same thing that we use the alkaline for the degreaser, which we're going to use most of the time, and the descaler or the acid for the scaling part of the equation. But most people, I find that when they talk about using soaps, they find that they haven't gotten the results of the soap. But normally it's because they actually don't follow the proper procedures with soap. So I made up 4 rules to follow. They're quite easy to follow. We can see that a lot of the tests out there showing that the soap is very efficient to use, even including the cost and time and product to use. So if we go into the rules, the first rule is to dilute according to label. That sounds pretty simple. But as you can see, there's not very many different labels that are out there, and the percentages are here following it. And if you're using a tank like that where you don't add extra water, it's quite easy. You can just follow exactly what's on the label, mix the tank at the right rate, and you can apply it to the wall. But the problem is a lot of guys use an inline injector or a foam gun or a injector right on the pumps themselves or a hydrofoamer. And these systems work by the water pressure actually drawing a little bit from a stock solution to produce the product outside. And that product has to equal what our label wants us to reach the wall at. In order to measure how to concentrate your stock, it's easy to do. You take 1 liter of a product and you start your system up and you spray into a bigger container. Once you have that bigger container amount, you just times it by the rate that you want to hit the wall at, and that is easy how you can figure out exactly how to concentrate your stock. You can actually go to our website, it has it there, where I actually have videos actually how to test that procedure I just showed you, and actually charts that actually you can put the numbers in to actually find out exactly how much you need to do and how to concentrate your stock solution. Rule number 2 is to apply with low pressure. And this is so that it actually will stick to the deposit that you want to clean. There's 2 main ways we can apply. We can use foaming, which is great because it's low pressure and the adding of air, which is really good for very smooth surfaces because the air actually helps the product slow from dripping off the surface. However, spraying is another option, especially when you have raw woods or open cement and stuff like that, because sometimes when the foam is put on, the bubbles actually hold the product out of the pores and don't get the penetration as well. The third rule is give it time to work. As you can see, these deposits are actually ripped apart and put into what's called a myocell. So that the soap actually surrounds the deposit and holds it up. And that takes a little bit of time. So if you have a real stubborn stain or you don't have much time, lots of time the best way to do is actually use a manual stimulation that helps the myocells form a lot quicker. The fourth rule, which probably was one of the most forgotten rules of all, is that you need to rinse the solution off before it dries. And especially when we're talking these big greenhouses that you have a huge amount of area, it's very important to do it in sections, to soak and wash in sections, rinse it off, and then continue on. Because what happens is that these deposits actually have to be floating in water. If they're not floating in water, they actually break apart and deposit back down to the surface, which doesn't help you at all. It actually puts the biofilm back onto back to where it was. So it's very important to rinse it off before it dries. So even though we did a huge amount of cleaning and most of the microbes are gone, there's still enough to become harmful for plants. So we need to use a proper disinfectant in order to get a good clean, a good final disinfection and control of the pathogens. The disinfectant really depends on a lot of different things. Number one, the type and cleanliness of the surface. Well, that I hope you did really well with your proper using of detergents. And then, of course, what type or resistance to which microbes you're really challenging with. So if we look at a virus, for an example, here's a bunch of different layers that viruses could have. The phospholipids or the proteins or so on and so forth. Not all viruses have all these layers, but they do have a very varying amount of each one of these layers, and some have more, some have less, some don't have certain parts of them. But once we know this, we can actually look at viruses microscopically and tell where they would fit in a group. And this is a chart of basically fixing those outer shells and telling you what groups, the envelope, meaning they got more fat on them, a non-envelope, we got more protein. And we rate them in this way, and it's really nice to rate them in this way because these are easier to kill on the top and these are harder to kill on the bottom. So that helps us be able to see how hard a particular one is. For example, the Rugus virus is actually really down here at the bottom, which is actually a very tough virus to get control of. So we go back to these protein layers. There's two different ways that proper disinfectant can kill. I call them the stuffers and then the rippers. And basically the difference is that the stuffers, what they do is they actually block a absorption site for food for the virus or bacteria, and also block the excretion site. So they can't get rid of their waste or can't eat anything else. So they basically suffocate the pathogen. Whereas the rippers actually rip apart the wall and let the DNA and RNA leak out, which actually kill the pathogen, whatever it happened to be. So those are two different ways that we see that it can kill. So now we can take, now that we know there's two different types of disinfectants out there, and with all the other things, we can actually rate them on our list. So if we take an example, these are our example of our products that are out there, quaternary ammonias, and then these are glutaraldehyde quaternary mixes. If you're not sure what you're using right now, you can actually send me a link or question, and I can actually help you which one it sits on. But these first three are actually what we call the blockers. And the blockers are actually very temperature dependent. And what I mean by temperature dependent is that if they're all measured, their labels and their rates, what they can kill, are measured at 20 degrees Celsius. If the surface, the water, all the different environments that are all that when you attack the surface with the specific disinfectant is only at 10 degrees Celsius, you may have to double the concentration or double the contact time to get the same result that you would have had at 20 degrees Celsius. Whereas it doesn't matter for the the rippers or the oxidants, where the time doesn't matter. I'm sorry, the time is a lot quicker because they just have to rip the wall, but also that the temperature doesn't make a difference. So a couple different things about applying disinfectant. I have this question very often. It's actually very, very important to prevent over dilution by actually letting the surface dry before you actually put the disinfectant on. That's why I recommend to wash, use the soap, clean, do it in sections. At the end, just spray and disinfect the whole entire area at once. It also prevents any incompatibilities, if there is happen to be any, between the detergent and the disinfectant. Improves the penetration on those pores we talked about earlier. And of course, we got to do just like we did with the soaps, apply according to label is very important. And let the disinfectant set for at least 10 minutes to do its proper work. Secondary disinfection, this is the fogging. It doesn't replace the actual liquid disinfectant, which we just talked about a little bit earlier. The second disinfection, the fogging itself really relies on temperature, relative humidity, room saturation rate. All those kind of things are very important when you do it to make sure you have a great downtime before you enter, and ensure you're going to have a great downtime before you enter the greenhouse. So it's actually, this is a secondary, it's just an extra thing to get in those little crooks and crannies you couldn't get with the liquid disinfection. So it's a secondary disinfection process. Now that you've done all of that, it's really important that you protect all the stuff that you worked and cleaned and worked at. And I like to talk about using this is called a Danish entry protocol, is to use clean or disposable protective gloves when you go into a greenhouse. And basically the way it works is this A side is basically the outside of your greenhouse. D is on the inside of your greenhouse. C is what we call the critical control point, or an area that you try and stop anything from crossing the barrier. It could be as simple as a line on the floor or a bench or a wall or whatever it happens to be, something that stops somebody from coming in from outside and walking directly into your greenhouse. At that point, you might want them to change outerwear that might bring in possible soils or pathogens from outside, change footwear if possible again. If you don't know how to change footwear, some people like to use foot baths. Foot baths are one way to help stop pathogens coming from one side to the other side. I myself prefer to use more of a spray system. These foot baths get dirty really, really quickly, and the pathogens are challenged in the bath itself, and of course are not nearly as effective on the second boot that goes in there, and third and fourth, and then how many times till you actually change it. Whereas you use some sort of a spraying system, whether it's a hand sprayer or a garden sprayer, you can clean your boots and then spray your boots with a fresh brine of disinfection, so that you can you get the best effect on whatever happens to be left on your boots at that time. So biosecurity itself, like I said earlier, is a huge, huge area. I only covered this one little tiny area here. There's a lot more things to talk about, and I'm sure we're going to be talking about them in all the presentations that come up later. So I want you to be aware about all the different things. And I think one thing that I've learned this year with this COVID is we learned how important the detergents are and how important biosecurity and the living science of biosecurity, how we're learning different things all the time. How important it is to keep up on the science of the whole procedure. So that's all I have for you today. I know it was really quick, but I just thought it would be good to review what we talked about. Karen?
That's great. Thanks so much, Dave. What a great presentation. So I'm just going to have a peek here to see if we have any questions yet. Sometimes they take a minute to show up, but so I don't see anything quite yet. So I have a question for you. So oftentimes our cleanouts are done, at the end of the year for conventional crop cycles, right? So there are, in my area anyways, the temperatures can be pretty chilly at that time. And so I think it's a hard sell when you don't have a crop in um, to heat the greenhouse during clean out. So I'm just curious, if you can comment on how important that is for the efficacy of disinfectants and clean out in general, to have warmer temperatures as opposed to cooler temperatures.
For sure. If we look at the detergents first, detergents is actually um, cold water is the hardest to wash with. Warm water is better or easier to wash with, but actually cold water or soap is more important. So soap is actually more important than that. But the one thing about soap with water is that you don't want to go over 140 degrees Celsius or sorry, Fahrenheit. Or 6 degrees Celsius is too actually hot and actually bakes protein. So keeping your greenhouse at actually a room temperature is actually the ideal way. But in soaps, the heat does help. As for disinfectants, we want to try and keep them as close to room temperature as possible. But if you are using colder temperatures and use one of those blockers we talked about, you're going to have to increase the concentration to get the proper amount on there. So it's a give and take. Whatever you have, it costs more to heat the greenhouse or to use a little bit more. Product. I guess that's a balance we have to look at.
Right. Okay. Thank you. So there are some questions that have shown up, but one in particular is asking if you can put the web link back up for a minute. So I wonder, is it possible to re-share your screen?
Yep. Right there is the web link on the bottom.
Okay. Okay, so maybe we'll leave that up for a minute while I ask you this next question. So for grow tents, do you have some comments on cleaning them? Would they keep the same procedures as a greenhouse if they work with sturdy grow tents?
Sorry, I'm not understanding the question. Grow tents?
Mm-hmm.
What do you mean? Oh, grow out like outside tents?
Yeah.
Yeah. Yes, they would actually, because it doesn't matter where the pathogen in, it'll still be on your grow tents as much as inside the greenhouse. So yes.
Okay. Okay. So it looks like another one here. So what cleaning regimen would you recommend for a permanent, mostly tropical collection?
The nice thing about a tropical collection is you probably get a little more heat in there all the time, which is very advantageous, especially with washing and applying disinfectant. But the procedures are exactly the same. The proper use of soaps, especially in a tropical one, you'll have a lot more organics going. So you're going to spend a lot more time with your alkaline detergents than you would with your acids. So, but yes, the same regimen, and again, you can find it on the website, the actual protocol.
Great. Okay. And I think that is all the questions we have for the moment. But Dave, I understand that you're sticking around to the end.
I will.
Yeah, so if any other questions come up, please feel free to type them in and we can always ask them at the end of the session. So thanks again, Dave, for a great presentation. and we'll see you shortly.